So on behalf of Dr. Lynn Malik, who's an associate professor of medicine and pediatrics at Versity Blood Center, Medical College, Wisconsin, and myself at Emory University, our task today is to talk about technologies and advancements in inhibitor treatment. And, I was thinking about this topic. If this talk was given 7-8 years ago, there'd be no advancements. It'd be the same talk probably for the last 10 years, but there have been some advancements and some reconsiderations, and we're going to kind of split up the talks halfway through. So we'll go ahead and get started and hopefully have enough time at the end for questions. All right, so these are objectives. We're just going to try to get a sort of a basic understanding of where we've been and where we are. We're not going to spend too much time on the past because there hasn't been a lot of improvements. We don't need to spend a lot of time talking about that. We're going to talk about what's going on in the inhibitor world and sort of things that are sort of on the horizon. This isn't going to be a discussion of all the novel therapies that are on the way because we've already had that talk multiple times during the session. Really try to talk about what we're doing now. So let's talk a little bit about prior and current treatments. And so many of you seen this sort of stepwise approach where we've been. There was a nice, in the science fair, you can sort of see where we've been in progress over the years. And so, inhibitor management really didn't come into effect until the 1980s and 1990s. We started to develop bypassing agents that allowed us to give therapies for patients with inhibitors. Prior to that, there really were nothing. We've added extended half-life products for hemophilia care, and certainly the most disruptive and most impactful drug that's come on the market right now is emicizumab. I don't think anybody, even I think the company, would not have predicted that nearly 90-something% of the patients on inhibitors would be with inhibitors would be on emicizumab. So I think that's been a huge impact. And at the time when emicizumab came out, a lot of us were wondering, well, what are we going to do about those with inhibitors? And so just sort of as a background here, we know that antibodies or inhibitors are unfortunately common. They're at least 30-40%, particularly most affected in severe hemophilia A. And somewhere in depending what you read, this as high as 8 to 9% in severe hem B. I think it's maybe an overestimate from the PEDNet data. But, when someone develops an inhibitor, there's a huge impact on the quality of life. You can't use the things that you normally would have used to treat and replace factor treatment. There's significant orthopedic issues. Even patients on emicizumab, long-term, they're going to have problems with orthopedic issues. It's not going to be the same as it was before, but certainly it's going to continue to happen. And so I think we need to think about, if we're able to tolerize patients, we should really consider it. One of the things I got really nervous about is when emicizumab came, people just said, oh, what's the point of ITI? What's the point of tolerance? I think that's was a little short-sighted. Thankfully, all the organizations have come around to the idea that it makes sense to do this. And so just briefly, we have, available therapies. The efficacy, one of the things that was really interesting is that once emicizumab came around, we realized that the efficacy of these products were not very good if you didn't have good hemostatic control. It would take routinely 15, 20 doses to treat a joint bleed with either bypassing agent. There were all kinds of issues with it. The volume was pretty outrageous. We had patients on fiber prophylaxis that were getting, 100 cc's a day of, their factor treatment for ITI, and then they would get 100 cc's of fiber. I mean, they'd be sitting there for hours getting their treatments. One of the things that we really wanted to make sure that people understand, and I think this isn't really fully utilized, is that if somebody has a low titer inhibitor, you should consider giving high dose factor. And it's sort of obnoxious to me to see people getting bypassing agents when they have a Bethesda titer of like 1. There are many situations where you could give factor 8 in that situation. And maybe sometimes we want to save it for other situations. The way that we really determine how well it works is that you check a level 15 to 30 minutes later. If it takes 100 units per kilo, if it takes 200 units per kilo, if you get any recovery, 40, 50%, that's going to be better than any bypassing agent, always. And people need to understand that. And we've done pretty tremendously high doses of factor 8s for those patients with sort of low titer inhibitors. We do have Obizur available. It's not indicated for use with congenital hemophilia with inhibitors, but all of us as providers have it as our backup plan in case something happens with a patient that we just cannot get them to stop bleeding. We're going to show this chart a few times, so I'm not going to spend a lot of time on it. Obviously there are lots of choices that we have for bypassing agents. FIBA has been around a long time. The dosing listed here is for those not on Hemlibra. Of course, you have to be very careful limiting the dosing to 100 units per kilo in a 24-hour period for those patients. The effect, we typically use FIBA a lot in the hospital, significantly less expensive than Novo-7. I don't think that's a dig at Novo-7. I think they're well aware of that. It is a much larger volume. If a patient's already on IV, we typically would use that in the hospital. It worked very well, for prophylaxis. It depended, Novo-7 prophylaxis worked very well, but you had to give it almost every day. And that was quite challenging for a lot of patients. It was recombinant. It had a much smaller volume, could be given much faster. So they both had their benefits, but it definitely was a struggle. And then entered a new field. And so previously, the generic is called Epticog Alpha, and this is called Epticog Beta. And so a new bypassing agent has entered the market, and it's something that's starting to be implemented. And we'll talk a little bit about this in upcoming slides. And MASAC has come up with the recommendation because the trials did not include emicizumab. It happened before patients were, all on emicizumab. And so most of the data we have is on that. But we've come out at MASAC saying you should consider using the lower dose regimen. And we'll talk about that lower dose regimen in a minute. There have been a number of Haven trials. There's been even more. We have Haven 6 that's completed as well. But most of these patients on this trial were well controlled. The majority had no bleeds, and majority were traumatic bleeds. And just as a reminder, people were not, there has never been any evidence of bleeding following physical activity. I think there's a lot of discussion about that. And we don't see that. It was never seen in the trials. We know that it was pretty widely successful. And there are going to be a few patients here and there that don't respond as well. But I think overall as a group, it was wildly successul and works very well for most patients, particularly those with high titer inhibitors. We know the mechanism of action here. I'm not going to spend a lot of time on this, but really it's important to understand it is the standard of care for persons with hemophilia A and inhibitors. It should be offered if you have a high titer with inhibitor. It's fine if you don't want to consider it as an option, but it should be offered. It is the standard of care in the United States and in the Western world. And, other options certainly can be used, but there's there hasn't been seen any benefit of those other options. We'll talk a little bit about those other options. And so there are a few of you that, aren't on the Hemlibra train, and that's okay. There are other options, and we talked about these just a minute ago. You have APCCs or FIBA or FIBA, depending what part of country you're from. These are options. They do work. Prophylaxis is something you can consider. And again, the chart here, looking at illustrating the differences in the doses. I'm not going to spend time on this, but just again to illustrate that there are differences in the doses, there are differences in the frequencies, and that's important for people to understand. If you look here, the MASAC recommendations, you can dim the lights. It's kind of, I don't know if that's a little hard to see in here. Is it possible to dim the lights, or? I can't even see it, so. Yeah, so, let's see if they can get the lights down a little bit here. So I wish it shows a different color here. But the point of this is that the MASAC has recommended that prophy with bypassing agents should be considered in those with inhibitors. This obviously preceded all of our discussions about Hemlibra, but there's really no clear-cut guidelines when to stop it. The dosing regimens are listed here. FIBA prophylaxis can be used, and we wanted to include hemophilia B on here as well. You see the dosing on here, and these are older recommendations from MASAC, which is actually going on right now. So wanted to talk, just because it's a new product, I don't want to, say we're preferentially saying, Epticog Beta over Epticog Alpha, but Epticog Alpha has been a very long time. Many of you have been into inhibitor summits. They've talked about it. There hasn't been any new data on it, so we're not going to talk about it. But there is a new product, just something to consider. It's called the PERCEPT program. It's always these very tortured acronyms here, but the Program for Evaluation of Recombinant Factor VII Efficacy by Prospective Clinical Trial. And so there were a number of trials that were done. You can see here there was a dose range study. There's the Percept-1 study, which we're going to talk about. That's the pivotal phase 3 trial. There's the pediatric trial, which is the Percept-2, and then there's a surgical trial here. So we're going to talk just about this one very briefly, but you can see there are 27 patients and 468 bleeding episodes. And so you can see that there are quite a number of bleeding episodes to get some good experience. And this was a crossover study. There were 2 regimens that were chosen, and for the sake of time, we're just going to summarize it here. There were 2 doses. So there was the 225 micrograms per kilo. You gave that dose, and if you needed another dose 9 hours later, you started giving 75 micrograms per kilo. There was also a regimen in which you gave the lower dose, 75 micrograms per kilo, every 3 hours, and you continued it till you had good bleed control. But what we found is that, and I think it's okay. I think what we found was that this dosing was highly efficacious. The vast majority of patients, remember, these are patients with inhibitors without being on Hemlibra, were able to be controlled with 1 dose. I think in trials, we often do that because we have to evaluate. In real life, we've used this, and oftentimes we've given maybe 1 more dose just to ensure they don't have that rebleeding issue, which I don't think is a problem. They're still investigating whether there's some sort of difference, whether it's a biosimilar or not. It does have an increased affinity for the endothelial protein C receptor that may convey some additional hemostasis, but we'll have to see what happens out in the real world. But just something to be aware of, something that's another option for those patients on emicizumab. And of course, it's also efficacious for those with hemophilia B inhibitors. So one of the questions that comes up a lot is about low-dose APCCs, and we've utilized this at our center, and we've actually utilized it at outside centers where we've managed a patient. And we actually had a patient that had a skull fracture. He was on emicizumab and developed a skull fracture, had gotten multiple doses of recombinant 7A. After about 3 or 4 days, was not having control of bleeding. And ultimately, they made a decision. I think they made the only way they would do it is that they put my name on the charts. The doctor said, and they said it's okay to give this dose of APCCs, because during the trial we were very afraid of the events that developed during the trial series, in which patients developed thrombotic events. They developed a phenomena called thrombotic microangiopathy. But after really evaluating it, it really only happened in those that received sustained doses and doses over 100 units per kilo. And we've utilized this lower dose regimen and have not seen this at all. And there's a potential that this may actually work better than factor 7A. But that patient actually had a pretty brisk response within 24 hours and was able to be discharged. And this was outside of our institution, but somewhere we managed it. So I think it remains a option if or when recombinant 7A fails. So we like to have a lot of backup plans in case things go wrong, because usually that's when I'm getting called by somebody when they've tried multiple different things, and we want to make sure we have good backup plans. So one of the things, and I'm not going to read everything on here, but this study should be published soon. This is a study conducted by Hyundai and Guy Young. And what they wanted to do is they wanted to look at this concept. People forget on the clinical trials and on the Hemlibra clinical trials, there was a child that had that appendectomy and got a single dose of FIBA and did totally fine. There were no issues related to it. Potentially, if he'd got multiple doses, there could have been issues. And so, there is this idea that low-dose APCCs potentially could be done. And so what they did is they took 9 patients with severe heme with inhibitors. They were on emicizumab, and they had 2 different arms. They wanted to collect blood samples, and they wanted to do something called thrombin generation. They got multiple escalating doses. And so you can get their samples. You can spike in higher doses of APCCs. And then you can also give the dose in real life. So you can do it with the sample, or you can give it in real life and check it. And they wanted to see, was there going to be any kind of discrepancy in that? And they did. So if you look on the left-hand side, this red part here is actual. So this is like in real life, and this is in vitro. So this is where they took their sample and they added emicizumab, or they added APCC to it. So what this just shows is that, a lot of the stuff that we sort of understood and came out of the Hemlibra trial series, that giving those doses of APCCs causes exaggerated thrombin generation, We weren't seeing that as much in vivo. And so, there's some question about how these samples were collected, but certainly there's a big discrepancy. And this is where you have to be very careful when you do things in the lab without humans versus doing it in real life. They don't always match up. And so, not to be outdone, or always trying to outdo Guy Young, we're trying to conduct a similar study at our institution. And we're actually going to have patients use the low-dose APCCs moving forward. And I'm not going to read all this, but the primary objective is really to look at the safety of using low-dose APCCs. We're going to have spiked-in samples. And what we're going to do is we're going to have patients give those doses and evaluate this. We have a couple of patients that utilize it. And that's okay. So I think at this point, this is the point where Dr. Malik's going to take over, and I'll be back in a few minutes.
All right. And the lights are going to go up and down while we're talking about this. So now we're going to switch to talk about inhibitor eradication and immune tolerance induction, and talk again, focusing on what some of the kind of newest data would be in this arena. So I think, as Dr. Sidonio already mentioned, as hemophilia treaters, we do think that it's still important to consider attempts at getting rid of inhibitors if you're able to. And some of kind of what we look at to think about this is really, how did patients on emicizumab do? What are some of the signals that we should think about in terms of still attempting tolerance? So I think importantly, if we don't attempt tolerance, this would mean that patients lifelong would need to rely on the bypassing agents that Dr. Sidonio has outlined. And we know in many cases, these do not provide optimal bleed protection, nor do they provide optimal bleed control if a patient has a bleed that needs to be treated or if they go to a surgery. And then the other piece that we wanted to highlight is just also the possibility of patients developing anti-drug antibodies. So these would be antibodies, the immune system recognizing the emicizumab, so not the same as the factor VIII inhibitor. And in the collection of studies that we outlined here, approximately 2.5% to 3% of patients who are on emicizumab can develop antibodies to that. The figure that's on the right-hand side of the slide is some of what had been studied in a patient, where they were looking in the black dots of what their PTT was. So when they take blood and put it in a test tube and ask it to form a clot over time when a patient was on emicizumab. And then this patient in particular started to have bleeds, which are outlined there in yellow. And when they surveilled the patient, they actually realized now the PTT is no longer normal, so reflective of the hemophilia. And what they were able to determine was that the patient's immune system had recognized the emicizumab. So again, really just calling to attention that emicizumab, like any other drug, isn't perfect. And things to consider are also the possibility of anti-drug antibodies. We will talk, as Dr. Sidonio mentioned, there are studies moving forward that look at using emicizumab also at the time of inhibitor eradication, and we'll go into some of the published information as well as some of the studies that Dr. Sidonio is helping lead from his center. This slide outlines some of the rationale about factor VIII prophylaxis. So some of the studies that have been done before the era of emicizumab really looked at what would be the optimal way to introduce factor VIII for babies or for pups to lessen the inhibitor rates. And so in a German cohort where they looked at escalating dose of prophylaxis rather than full prophylaxis, so 3 or 4 times a week, they saw that when factor VIII was introduced at those lower levels, they had a reduction in inhibitors. That being said, before the era of emicizumab, usually when we were talking about starting prophylaxis, once it started, we had a shorter time to reaching those kind of critical number of exposure days or doses of factor. In this slide, it really depicts a patient on the far left that would have hemophilia A without an inhibitor. They start their medication. On the top, you can see these would be patients, or maybe you can't see totally in the back, but in the lighter blue, these would be patients that don't go on to have inhibitors, versus those that are in the darker blue, either those that develop a low titer inhibitor or a high titer inhibitor. And then as we kind of cascade through the slide, patients undergo ITI and they either have successful ITI or they don't. So when we think about kind of historical factor VIII exposure, patients that are receiving factor VIII exposures kind of historically are thought to reach those 20 exposure days of factor VIII in about 4 months. That being said, when we do some of the modeling of when patients would have bleeding events after they've started emicizumab, this rather than months is going to likely be years. So meaning for those patients, there will be much more sporadic use of factor VIII, spanning many, many more years. And quite truthfully, as a hemophilia community, we don't totally know what that means for especially young patients that haven't had factor VIII exposure. So again, looking at some of this modeling for patients that started on emicizumab for prophylaxis, we would estimate that they would reach those 20 exposure days rather than with factor VIII exposure in 4 months, that this may be a decade or more. So some of the modeling thinking that this may be 13 or more years before they reach that. And so, you know, as a clinician, when patients come to clinic and we're doing inhibitor surveillance for patients that don't have inhibitors, this is, you know, that question for patients and for providers. When did you have your factor VIII exposure, and is there any risk of you developing an inhibitor, which likely would be different in the emicizumab era? So now thinking about kind of why would we do ITI with emicizumab? And as Dr. Sidonio mentioned, I think when emicizumab first came to market, there was talk about should we even ever attempt ITI, because for those of you that have been through ITI, it's really hard work to do ITI. It's very costly. It's really time intensive. It takes a lot for families and for patients to be able to undergo ITI. And I think that's something that we all recognize. But the reasons that we think about this is that even with emicizumab, patients still have bleeding. And so when we look at the Haven data, again, bleed protection is not perfect. And then as Dr. Sidonio mentioned, those aspects of what bleed control is like for surgeries, or if you have a bleed when you need to use a bypassing agent instead of factor VIII. And again, we recognize that that's not perfect. So when we think about surgeries, again, this would be a reason to think about attempts at tolerizing or getting your inhibitor to go away. Also in the middle of the slide, access to currently likely to come to market is maybe how I should say it, gene therapy, kind of late phase trials. So the ones that would be likely submitted to the FDA. Currently, those restrict patients that have had prior inhibitors, but we'll talk about some of the current studies that are underway. And then the last piece would be, you know, thinking about safety. I think as a community, we've had more and more experience now with using emicizumab, but certainly there are some of those safety concerns. So thinking about can you get rid of an inhibitor, I think is still an important consideration. So kind of drilling down into the kind of why is that an important consideration? We know that our inhibitors are associated with an increased risk of bleeding as compared to patients with hemophilia that don't have inhibitors, an increased risk of joint damage long-term, decreased quality of life. And some of this, again, goes into the aspects of just how kind of treatment intensive therapies are when you have an inhibitor. Cost of care, so the high costs of healthcare, this could be increased needs for hospitalizations as compared to somebody that doesn't have an inhibitor, as well as increased risk of mortality. So when we think about how do we eradicate an inhibitor, this is where our data really surrounds the use of ITI, immune tolerance induction. So this is a recommended treatment option for inhibitor patients, and we'll talk about some of the aspect of patients with new inhibitors versus existing inhibitors and what some of the recent publications have shown. We have lots of experience with ITI, so this has been a way to treat patients for over 40 years. And overall, we have about a 60 to 80% success rate. So although this is really hard work and it's very costly, we do have a better chance of success than failure, thankfully. So many of you may be familiar with what are the kind of standard or historical ITI therapies. They're listed here, the Bond protocol, Malmo protocol, the Dutch protocol. All of these different protocols really use different intensity of treatment of factor VIII. Some of them included other medications to really kind of target the immune system and do some what we call immune modulation. And now that we have emicizumab available, and with Dr. Sidonio's group really leading the way with us thinking about this, is there are also now other regimens of ITI that use emicizumab in the background. And the idea there is that you would be using your emicizumab to control your bleeds and then receiving factor VIII therapy to try to tolerize your inhibitor. Dr. Sidonio will talk more specifically about the Atlanta protocol. So this is a protocol that he developed with his colleagues at Emory, using what we would consider kind of a mid-range dose of factor VIII. For ITI, historically, low dose was 50 units per kilo, high dose 200 units per kilo. With the Atlanta protocol, you use 100 units per kilo 3 times a week. In the low dose protocols, that's a similar interval of when you would give factor VIII exposure. In the high dose protocols, it's typically daily. And patients also receive emicizumab. The specifics of that, I will just kind of call out that the emicizumab is used really in standard fashion. So the same way for any of you that are on this, the same sort of loading regimen and then continuation or maintenance regimen after that. So when we think then about the Atlanta protocol, again, Dr. Sidonio and his colleagues really presented, I believe it was at an ASH meeting initially, kind of the aspects of the Atlanta protocol and some of what they were doing at his center. So when they looked at some of their initial patients, so this was 7 patients who were receiving that Atlanta protocol, so factor VIII for ITI as well as emicizumab, you can see some of the data here regarding patients, specific patients and their inhibitor titers over time. So of their group, and again, this was some of the first published data in this area, they had 3 patients that were able to get rid of their inhibitors, 2 patients who achieved a normal factor VIII recovery, meaning when we give you a dose of factor VIII, your level goes where we would expect it to. They had 9 bleeding episodes in 4 of the patients, but importantly, they saw no significant safety signal. So some of those concerns about clotting or thrombotic complications, they hadn't seen that in their group. So I think with this in mind, you know, as a hemophilia community, we've needed to think about, well, how do we standardize some of this so that if you were coming to my center or Dr. Sidonio's center or a center, you know, in your home state or city, kind of how do we optimize some of your management? So there was a group of clinicians from really across the globe that came together to talk about some of the factor VIII tolerance. They called themselves the FIT group. This is probably somewhat hard to read in the back, but they came up with a treatment algorithm of kind of what doses you should start at, when you should surveil patients, when you should think about maybe making a treatment change or even stopping ITI. So here we have on the left, this would be a patient with a new inhibitor. They recommend a similar but not quite the same dosing as the Atlanta protocol. So they recommend a low dose protocol of 50 units per kilo 3 times a week with emicizumab, and then really reevaluating kind of the aspects of inhibitor eradication. And for patients that are moving kind of through this treatment, really thinking about, would we make other modifications? So bump up your dose, or consider even potentially, if you're not showing any evidence that your inhibitor is responding, to think about discontinuation. So, you know, I think something that, you know, Dr. Sidonio and I are very passionate about is really making sure that, you know, with hemophilia being a rare disease and with inhibitors being a rare complication in that rare disease, that we really need to work together as a community to try to standardize what we're doing so that patients have a best outcome. So now thinking about some of the newer studies, there was a study that the information was, again, presented at ASH this year using a lactate, which is a recombinant factor VIII product that has a half-life extension by fusion to the Fc portion of a immunoglobulin or an antibody. In this study, these were patients called the Verity-8 study who had newly diagnosed inhibitors or inhibitors that may have been diagnosed a while ago but didn't ever previously go on to ITI. So we thought of this as first course ITI. These patients did not receive emicizumab, just the extended half-life product. And they received dosing at that high dose regimen, so 200 units per kilo every day. And what we saw with this study is that using this specific drug, which there's been some thought about, does this newer drug at all interface with the immune system in a different way that could provide different benefit? What we saw, and this is a plot of the percentage of patients that were tolerized here, and then, the time to tolerization. And what was really noted is that when it worked very fast. So on average, patients achieved tolerance on the order of 12 weeks from when they started. So this is very different than some of the historical pieces where we would go, not just weeks, but years. We may go up to 36 months. So in this cohort, when it worked well. And kind of the overall success rate that you can see there was 67%. So kind of in line with what we had seen historically. Some of the advantage this may present, again, is that time to tolerance, so that you're not on that ITI regimen, which is very hard work to do for quite so much time. Looking at the same medication, there was also a study called the Reiterate study. This was for patients that had previously failed ITI. So they had a different regimen utilized in the past, but still had an inhibitor. They also received that high dose factor, the aloctate, 200 units per kilo per day. Overall, it was a small number of patients, so 16 total patients enrolled. 7 of them withdrew from the study. The 9 that completed the full study, not quite half, 4 reached a negative inhibitor at some point in time, and that's what's depicted on the right-hand side of the graph. So again, these were patients that had already had ITI in the past, but went on to use this different medication. So now I'm going to turn it back over to Dr. Sidonio, and we're going to go through some of the future directions.
All right, so let's talk about current studies, future directions, as we round up the last 2 sections of this talk. So, we really wanted to, as mentioned before, when emicizumab, we were using it in the clinical trials, we had a couple of patients that didn't, we had one patient that got one bypassing agent, went directly into the trial. And during that trial, Dr. Meeks and I were thinking, would we ever try to tolerize this patient? He's doing very well, didn't have any bleeding, similar to the data. And, that family didn't know the previous inhibitor experience, which, was a nice thing for them. But also, we wonder, should, a toddler, should we just say, well, they're just going to have an inhibitor the rest of their life and we're just going to be okay with that? And I wasn't okay with that. And so out of that, we really wanted to come up with an idea of how do we get at this issue? What I didn't want to do is that we spent a whole decade trying to figure this out through retrospective studies. So we said we should do this as a prospective study. And we had a few questions. So this is the Atlanta study, and it's got a longer name, the emicizumab PUPS and NUVIC ITI study. It's a phase 3, 2-part prospective open-label multi-center study that's only done in the United States. We only have it open at our center. We've been trying to desperately get it open at other centers. Everybody is short-staffed right now, so research organizations are struggling. But part A of the study is going to focus on tolerizing patients, so hoping they can actually reduce inhibitors. And then part B is, what do you do after they get an inhibitor? So let's talk about part A here. So our question for this one is that, like Dr. Malik was mentioning, there was a study done by Baxter years ago, when it was Baxter, and they wanted to give low dose Advate to patients early on, very low exposures, have the nurses do it. Maybe you can sneak in the factor, trick the immune system to accepting it. Similar to what the thinking is about like peanuts and, introduction of like very low dose of peanuts. And the study failed, but it mainly failed because eventually hemophilia patients bleed, right? This is before emicizumab. And so when they bled, then they got high doses. And so that whole concept, it got blown up. But that German experience, and this is the important thing, that German experience when they did it showed tremendous benefit. They barely had any inhibitors for years afterwards. And then we all tried it, and the inhibitor rate was 40%. So it was no different. So this is always the danger of single center experiences. So we wanted to say, can we give low dose factor? Octapharma provided the product NUVIC. And can we sneak it in at low doses while you're on emicizumab? patients aren't going to bleed, and can you actually get them tolerized so that we can actually reduce the overall rate of inhibitors? And does that early exposure also prevent life-threatening bleeds? And so we're going to take 40 pups, less than 3 years of age, moderately severe hemophilia, and of course you can't have an inhibitor go into part A of the study. I think this is a question that's been going on right now. Once you start a baby on emicizumab, do you give them low dose exposure, or do you just let them have bleeds and then give factor as needed? And so that's a question that our community has. And so, so the starting part of it is you just start them on emicizumab. Within 6 months, you start the low dose factor exposure, and that's 25 units per kilo every 7 to 14 days. And so that's what we're doing. We're going to follow them for 50 exposures. Most patients after 50 exposures aren't going to develop an inhibitor. And so that should give us that answer. We're going to look at the incidence of inhibitors and look at a lot of safety endpoints, and hopefully look at subclasses of inhibitors and antibodies that develop. And so this is something I think is an important question our community needs to have. What I don't want to do is have some centers do one way, some centers do others. Then we try to analyze it, and there's a ton of bias in that, right? Because you pick patients on one trial because you think they might have inhibitors versus other. And so we want to do this in a rigorous fashion. And so again, we're going to be testing them fairly frequently. Once they start Nuvik, every 4 exposures, every 6, and then every 12 after a certain number of increments. We're trying to balance not being too obnoxious with lab draws. These are babies, of course. So what about part B? So part A is, can we tolerize patients to accept factor VIII is not a foreign protein? And then part B is, what do you do after they develop an inhibitor? So if they develop an inhibitor, or if they already have an inhibitor, we want to evaluate the Atlanta protocol in sort of a multi-center fashion. We wanted to use Nuvik because this is provided because we were worried about insurance covering this. So we're going to have 20 patients under the age of 21. They have to have some measurable inhibitor within a certain period of time of enrollment in the study. So I wasn't intending on having 2 studies open at the same time. They're answering similar questions, but the pandemic came, and so everything just is like whatever. So it happens. I think it's still good to have this one. So this is an international study. This is a study that was investigator initiated that I worked with Carmen Escarola Eddinghausen from Germany. We wanted to take patients of any age with inhibitors, and it doesn't matter how many attempts. So we wanted to really capture typical observational experience. So it's been deemed as observational. I think it was deemed as a pragmatic, practical trial in Germany, which I don't really know what that means. And we wanted to let the docs do whatever they wanted to do. You had 3 options. We're going to follow them for 5 years. We wanted to capture all the different approaches, document how people are responding. So, a lot of people aren't going to be able to go on the trial for many reasons. But if you started ITI recently and you want to go on Unmotivate, the only restriction is you have to use, we wanted to keep the same product because we were worried about very messy data if you had like 17 different factor products on the study. And so this is what it looks like. There are 3 groups. There's sort of the traditional ITI. And what's really interesting is that group is struggling to enroll in right now because when we started this study like 4 or 5 years ago, that was the standard of care. And it's shifted significantly to our Atlanta protocol. And so we see a lot of patients not being offered group 1. There's nothing wrong with group 1, but it's just not very popular as expected. Most people would rather start emicizumab and then add ITI at some point later. And then we wanted to have a control arm of those on emicizumab. And we know that patients may move from arm to arm. Somebody may start ITI, it doesn't work. Then they start emicizumab and they say, let's restart ITI. And then at some point they become tolerized and they can go to group 3 and we'll observe them. And so we're going to compare outcomes in group 1 and 2, and then we're going to compare bleed frequency. And the way we measure ITI success is fairly standard. You have to have a negative inhibitor titer with a good washout. You have to have a reasonable recovery. You can see the numbers there. And you have to have a half-life of at least 6 hours based on kinetic studies. And so we want to measure this. We actually should have some safety data early next year if things continue to go well on the way this is going here. And before I get to the next slide here, I think we have about 33 countries involved in this study. And we're struggling in certain parts of the world to get studies open, but we feel pretty confident in 2 years we're going to have pretty good data to show everybody. We'll have a little bit of data next year because we wanted to have 20 patients that at least had 1 year of data, so we didn't want to report too early interim analysis. So this study is open, I think, in 3 sites in the United States, and there are about 8 or 9 sites that hopefully they'll get their acts together and get their contracting done. So otherwise, they're going to keep getting emails from me. So, we talked about in other trials, in other talks, obviously this is what's going on now, and there will be a few more that Dr. Malik will talk about. But, there are other drugs and other things that are being considered and that may enter the realm and may be usable. Fitusiran may enter the market maybe in 2024 or so. That will be a good option. Potentially, most of the patients will be able to get a single injection once every 2 months. The rest will get it once every month. We're hopeful that some other products like Concizumab, the anti-TFPI drugs, there's a drug that affects the way protein C works. That drug is moving along as well. So there's a number of investigations that potentially could be useful in attacking inhibitors, but if they're still at least a year or 2 away. And so we don't want to get everybody's, I feel like we did that with gene therapy. Every meeting was like, oh, it's coming next year. It's coming next year. And we thought it was going to be here this year. But of course, it's not going to be. Likely Unicure hopefully will be approved later this year. We're hoping that BioMarin will be approved in the United States next year. It did get approval recently in Europe, so we're going to see what happens in Europe. So I think this is the part where Dr. Malik's going to wrap it up for us, and then we'll have some time for questions.
Right, and I totally agree. So we're going to focus on gene therapy, which I think most you have heard lots about, but really to think about what this means for patients that have current or prior inhibitors. So I think it's important to recognize that the late phase, so when we talk about phase 3 studies that have data where companies would be able to go to the FDA and ask for an approval of a product, the current late phase AAV-based gene therapies, so the ones that are likely to come to market, have excluded patients with a history of an inhibitor. And this is an ever inhibitor. So I have patients that were interested in participating in the studies, and they may have had a transient low titer inhibitor. They never even needed to do ITI when they were 2. Those patients would still be excluded from the study. So I think the soon to market, hopefully soon to market gene therapies won't be available for patients that have inhibitors. That doesn't mean that that won't be on the horizon. And so that's some of what we wanted to talk about. So one of the aspects in terms of the, we'll show on the subsequent slide, is kind of the next iteration of some of the company studies will include patients who have inhibitors. There's also other technologies. So I think we are using kind of gene therapy, which is much more of an umbrella term currently to mean the AAV-based gene therapies, but there are other technologies or ways that gene therapy can be delivered that are being explored for patients with inhibitors. As a quick reminder, all of the clinical trials that are done need to be registered on clinicaltrials.gov. It's not the most user-friendly site to sift through, but you can go in and put in specifics of hemophilia A with inhibitors, hemophilia B with inhibitors, and just see what studies are there. And it will be studies that are not yet recruiting patients, so meaning not putting patients on study yet, studies that are open to recruitment, studies that are closed to recruitment. And then as part of the requirement after the registration is also that whoever was conducting the study needs to also update the clinicaltrials.gov website with some of the aspects of the findings of the study. So I think this is something that we do for our patients. I think, as treaters, we're pretty aware of the studies that are going on, but this is something that, I would encourage you to think about going to if you have some curiosity. Again, it's a little bit tricky to navigate, but I think once you get the hang of it, you can kind of filter what studies are available. So just highlighting, as Dr. Sidonio mentioned, hot off the press is the fact that the BioMarin gene therapy has been approved in Europe. This is something that we're still waiting for here in the US. The original BioMarin studies, as we mentioned, or the studies that completed late phase development, again, excluded inhibitor patients. But this is a study, it's called BioMarin 270-205. They're going to be recruiting 20 patients. These are all adult patients that have severe hemophilia A with a positive inhibitor in the last 12 months. Patients can still not have pre-existing AAV antibodies. So that's the antibody that is to the what we call the capsid, or really the shell of the vector. This is a phase 1/2 clinical study, so in early phase development. What they're really going to be looking at is adverse outcomes, but of course looking at also what are the trends of factor VIII. So this is a study that, again, I think we're encouraged in the inhibitor space to think about how patients will do with this therapy. Then a study that I wanted to also highlight that's from my own institution is different technology. As I mentioned, gene therapy is really that umbrella kind of terminology. This is a study that uses a different methodology, which I'll explain in a picture in the next slide. But this is a phase 1 study that's ongoing at the Medical College of Wisconsin for adults with severe hemophilia A who also have a history of an inhibitor. And the inhibitor could be an anytime inhibitor. It doesn't have to be a current inhibitor. It could have been a prior inhibitor. But patients need to be on prophy in order to enroll. The prophy could be of any type. So if you have a tolerized inhibitor, maybe you're on factor VIII prophylaxis. If you have a current inhibitor, maybe you're on emicizumab prophylaxis. So patients really come into the study on their prophylaxis. And then what is done, this is very different than the AAV-based gene therapy. That is, probably what you've heard a lot about, where it's a single IV infusion. With this technology, what happens is patients' stem cells, so those are the cells that are peripheral blood stem cells that are typically in kind of low volume in circulation. We actually stimulate those with medicine so that we can collect them, typically through an IV, or if patients have really good veins, through their veins. And then in the laboratory setting, those are then modified. And the way that they're modified, what is pictured here, is that your stem cell would basically be transduced by this lentiviral vector so that your megakaryocytes, they're the ones that give rise to your platelets, have your platelets actually produce your factor VIII. So this is not what happens normally, that your factor VIII is produced in your platelets. But the idea would be, if you have an injury, it's really that your platelets are there at the site of injury to start plugging up that hole and stopping a bleed. And so this is the idea that if your platelets contain factor VIII, then it's released at the site of injury. We have enrolled our first patient. It's a phase 1 study, so we're aiming to enroll 5 total subjects. You do travel to Milwaukee. This is a single center study. And I put up there just my contact information, and then this is the information for the lead doctor, Dr. Epen. A big difference compared to the AAV-based gene therapy, where you have a single IV infusion, is that you need to make room for these new cells that were, again, transduced in the lab. And in order to do that, patients have what we call pre-transplant conditioning. So they get medication, that's chemotherapy, to get rid of their old cells, and then they're delivered back their cells that were modified in the laboratory setting. Rather than it be outpatient therapy, patients are in the hospital. Kind of the thought is on average of 2 to 3 weeks. Our first patient that we dosed, he left, I think, at day 16. So he actually did very well. But so this is a very different technology. But again, just highlighting some of the innovation that really continues in the gene therapy space, including for patients with inhibitors. If anybody has any questions, Dr. Epen's very approachable. If you went on clinicaltrials.gov, her contact information is listed there. So she's happy to field questions, as would I. And then as we wrap up, this is really our last kind of full slide before we take questions. This is a slide probably very difficult to read from the back, but that's from a really great article that Mark Skinner authored, really talking about some of the aspirational goals in hemophilia. And so what we can see on kind of the greens in the green print is really kind of the patient relevant outcomes. So some aspects of really just trying not to have premature death. So before we had good products, kind of how do we keep patients from having life-threatening bleeds? And then really as we progress, our goals have really improved in terms of what things are achievable for patients that have hemophilia and patients that have hemophilia and inhibitors, to the point that we would really get to where patients have, optimal health and optimal ability to participate in sports or have activities that are, really physically challenging. And again, on the kind of therapeutic side, we've also continued to develop kind of new therapies that I think importantly are continuing to push the envelope. So this is an article that I think in a really great way is some of that aspirational piece for me as a treater, that we shouldn't just be focusing on, okay, how many bleeds did you have since you last came to clinic, but really, how is the therapy that we're able to provide you allowing you to live the life that you want to lead, and recognizing, I think, that every patient is different. So what one patient I see one week may be very different than a patient I see the next week, and having good therapies available, I think, thankfully allows us to tailor what patients' options are, specific to their personal desires. So before we take questions, we wanted to, we might have to put the lights down for this one. This is a picture of Robert and I when we were baby doctors. We couldn't figure out exactly when this was, but we think it was probably in 2010. Each of us had much less hair than we have now. But we have, both been committed to taking care of patients with hemophilia for a long time, and I think committed to innovation and collaboration. And so with that, nobody can take pictures of the slide for, holding us at ransom. But we'd be happy to answer any questions that you guys may have.
My question is, what about the inhibitor patients? The hemophilia B patients who are actually what about the inhibitor and hemophilia B patients that use ITI who have inhibitors who are your peds? Do you have any solutions to those patients that really, because that's what
that's a tough one. So there have been no advancements in hemophilia B inhibitor management in I don't know how many years, decades. So we've been using the same protocol, the Buto protocol. So there actually are no guidelines on hemophilia B inhibitor management just because they're exceedingly rare. The protocols that we have in place do work, but they're very difficult. So like we mentioned before, in hemophilia B inhibitors, about 5 to 8% of patients with severe hem B will develop it. The risk of hypersensitivity reactions are fairly high. They can range from just like a tickle in the throat to full-on anaphylaxis. And so when you do have hypersensitivity, we have to do a desensitization protocol. And oftentimes that means admission to the hospital and giving incrementally, starting off with very small doses and continue to give higher and higher doses until your body sort of accepts it without having a reaction. And so once you get to that point, and then you have to consider doing something called an ITI, and there's not really good data on this. Oftentimes it involves using IVIG. Sometimes people use steroids, and oftentimes it requires CellCept. The only person I know that has have documented some of that experience, there has been some data by Dr. Yasmin Abajas from UNC who's documented that experience. We have tolerized some patients from our institution doing the desensitization, and not everybody needs to do desensitization. That's only if you have a hypersensitivity reactions and can't get factor 9. And I don't know if you want to describe the case study that you did.
So we have a patient at our center who had a long-standing inhibitor and had been off of any sort of bypassing agents. He had developed issues with nephrotic syndrome, so issues with his kidneys with his ITI. But then unfortunately he had a bad bleed, a head bleed, and so we needed to put him on a treatment regimen. And we actually did ITI using the FC fused factor 9. So I mentioned the lactate piece is the FC fused factor 8. We used the FC fused factor 9. He did well. His inhibitor came back after he got factor 9, but then stayed away after we continued him on that drug long standing. We also, though, did the things that Dr. Sidonio mentioned, kind of upping the ante with the immune system. So we put him on other medications to try to get his inhibitor to stay away. He's done well, but I think, especially in the hemophilia B space being even more rare and the hemophilia B inhibitor space being even more rare, we have some appropriate caution in making broad recommendations when we have kind of a single patient at a center. But those are things to consider. The slide that Dr. Sidonio showed in terms of some of the innovations, thankfully, some of those drugs that aren't factor 8 or factor 9 really can be used in hemophilia A or hemophilia B with or without inhibitors. So that's where we will gain experience from kind of the hemophilia community, which is different than obviously historically where it was very much, these are A products, these are B products.
So there will be fitusiran, concizumab, one of those anti-TFPI drugs, will be able to be considered using in hemophilia B inhibitors, and they are enrolling patients in their trials specifically targeting that population.
So, excuse me, Chris and I are going to hammer the B questions here for a moment, but you guys kind of addressed some of the questions that I had, just some of the protocols that you've developed with newer technologies for A, and specifically like how can some of that transfer to B. But then the stem cell piece that you were talking about, is that something that you've thought about for B as well?
Yeah, so personally, I haven't developed the technology, but that is some of the ideas, kind of how can other blood cells be engineered to actually deliver factor 8, factor 9. So that is something that's being considered. When clinical trials come to be available at your center, there's always the animal work that's gone on in the background, the mouse work, the dog work, whatever it may be. And so that particular technology is much less in kind of in the works in the pipeline, but I still think that there would be hope for that for the future, because I totally agree. We need to keep up with moving the needle in the hemophilia B space, and especially the hemophilia B inhibitor space, because we haven't made the advances that we would have hoped.
We have one question online, and they're going to end in 2 minutes.
So the first question is, if factor 8 is used for a breakthrough bleed in patients on IMI, do we have to start induction of IMI doses again, or just continue the maintenance dose?
Yeah. So emicizumab and factor 8 are completely different. We didn't really get into this, but they don't have a synergistic effect. So when you give factor 8 when somebody's on emicizumab, the factor 8 is always going to outcompete. It's going to do its job. Its half-life is much shorter, so eventually it disappears, and then emicizumab takes over again. At some level, we're not really sure of, probably in the 20-30% level range. And so giving factor 8 while you're on emicizumab, you don't need to restart it. The beauty of emicizumab has extremely long half-life, and so it's very forgiving for missing doses. We don't want people to miss doses, but that happened on the trials, and there was no breakthrough bleeding events related to that. So,
thank you. And there's one additional question. Could you comment on the role of plasma-derived factor 8 versus recombinant factor 8 for ITI? Would you consider use of plasma-derived factor 8 for those who failed ITI with recombinant factor 8?
Yeah, another good question. I think the bottom line is that we don't have great granular kind of specific data in that arena. There's some of the idea with plasma-derived products leading to less inhibitor formation, kind of the flip side of the coin, inhibitors going away. We don't know. There is some signal, some centers, ours does not, but we'll switch patients to plasma-derived products for ITI. Again, we have not done that at our center as a practice, but there is some thought that the von Willebrand aspect of plasma-derived products may provide some benefit to immune tolerance induction.
and we didn't see any safety issues when we used, specifically we used alphanate in the Atlanta protocol. That's one of the first patients, and we didn't see any safety signals giving high doses of VWF with emicizumab. So,
yeah. Quick questions, please. For patients on emicizumab treatment and going for ITI, because to monitor the success of the ITI, we do the Bethesda assay, and usually with regular assay with the emicizumab, this is not indicative anymore. And in developing countries, they don't have the chromogenic assay. Is there a way to turn around for that specific for countries that they don't have the chromogenic, they just do the one-stage assay? Sure. How we can monitor this?
Yeah, so what he's referring to is, they have the chromogenic Bethesda assay. We have to use a special way of measuring inhibitors. It's not widely available in the United States. They can do it at the CDC, and they perform that for free for us, and probably do most of the inhibitor monitoring in the country. There are people trying to develop this. I know the Japanese group, and there's a group in Germany. And what they're trying to do is they're trying to find an agent that can neutralize emicizumab in the test tube. And that way you can actually measure it doing the standard measure. So you can measure factor VIII, you can do standard measurements. The issue with those, it's very hard to commercialize these. And oftentimes those groups say, well, we don't really want to spend the time and effort. It's just a headache to commercialize these assays. But I think there probably is a need for that, and something to consider, particularly as you get more, as they get a second generation with their 007 agent that they're working on. And then as Novo Nordisk has developing their first generation version of hemicizumab. Yeah, hopefully. So we are actually looking at it on the Motivate study. A German group has a neutralizing agent that we're hoping to evaluate. Yes, yeah, either that or Bond, I can't remember. Sure, go ahead.
Is there any way to predict which patients may develop the drug antibody to emicizumab?
To my knowledge, there isn't a great way to predict. So I think this is where, for us as clinicians, some of the story that I tell my patients is, if you start having bleeding events, we still want to know. Call our clinic. Let us know that you're having this as an issue. Mayo Clinic now offers, it's more of kind of a surrogate screen for the anti-drug antibodies. So it's not something that in our center we routinely screen patients for. But I think getting back to your question, we can't necessarily predict who may go on to develop those. So it's really important to just think about, if you're having bleeds that are new or different, or maybe don't make sense when you had good bleed control, that's a time where we would think about, do you have an anti-drug antibody? At our center, we haven't personally had any patients develop anti-drug antibodies, but again, it's something that we have looked for when the story is you have new bleeds.
Do we have one last question? We do have a session starting in here in 10 minutes. We got to wrap up.
Is there any more data about stacking emicizumab versus weekly, biweekly, or monthly in the number of breakthrough bleeding? I monitor a lot of Facebook and hemophilia sites, and I see a lot of people having more breakthrough bleeding when they're stacking than doing it weekly, because they're reaching their peak and falling farther than doing it weekly. I know there was data released a couple of years ago based on that, but has anything changed with that dosing regimen?
So there's no reason that anybody would have any different bleeding if they're on weekly or every other week. The way that monoclonal antibodies, it just doesn't make any sense. I've seen people say, well, I feel like I need it. It has a 28-day half-life. So it takes almost 6 months for it to completely leave your body. And so it's very unlikely you're going to see any differences. There's a slight possibility if you're giving it monthly that you might see a little bit more bleeding than weekly. But really, there's no signal on it. The levels are almost identical, so there's no reason to see any differences. And so sometimes I worry about some of this stuff. We didn't see it on the trial at all, and we haven't seen it on the post-approval studies at all. And so there's no reason to think that. I think practically we give it every week or every 2 weeks in most children, just because it's simpler in the volume and it's just easier to give, so it's not multiple injections.
Yeah, and I would just say real quickly, the initial data was that when you're doing it every 4 weeks, the actual concentration of the drug tails off a little when you're doing it every 4 weeks as compared to every 1 or 2. So our center hadn't participated in the Haven studies. So it was a new drug to us, to our patients. So we started almost everyone on every 1 or 2 weeks. I think when you do the load, that's the same dose as every 2 weeks. So we kind of naturally fell into a role of often just, this is the dose you're doing every week for 4 and then every other week. Now that we've had more experience, and for the patients that would like to go to monthly, we have switched them to monthly. And we haven't seen any increased bleeding with that. And I think as more data comes out, that slight trail in the plasma concentrations hasn't necessarily been associated with more bleeding. But I think my policy is always I listen to my patient. So if you feel like this is different and you're on every 2 or 4 weeks, we can change your regimen to something that, you feel more comfortable on, because we can't maybe always explain some of that achiness or things that are described.
Thank you so much.